Hello, everyone. My name is Jiaoyang Jiang. I'm a faculty member at University of Wisconsin-Madison. And our second speaker today, Adam Kositzke, is a senior graduate student in my lab. Adam obtained his bachelor degree in biology and chemistry from the University of Wisconsin-Oshkosh. He also, as an undergrad, he already elucidated a crystal structure of a bacteria, glycoside hydrolase, and that was quite impressive. And then after he joined my group, he developed an in-house galactosyl transferase assay to detect the recombinant and cellular protein O-GlcNAcylation. And then he also characterized the substrate characterization of the human O-GlcNAc transferase, OGT, towards the protein substrate O-GlcNAcase, that's OGA. And then so here today, I think he is going to talk about this project. There's the second one I mentioned about.
All right, today I'll be talking about a project where I've been elucidating the substrate binding mode of O-GlcNAc transferase towards O-GlcNAcase using electrophilic probes. So starting off with some general background on what O-GlcNAcylation is, it's a dynamic and reversible post-translational modification carried out by a single pair of enzymes, O-GlcNAc transferase, so OGT, which catalyzes the addition of the glucanac moiety onto substrate serine and threonine residues, while O-GlcNAcase, OGA, reverses this modification via hydrolysis. Now, over 4,000 human proteins are O-GlcNAc modified. Many of them are involved in different biological processes ranging from transcription, translation, signal transduction, mitochondrial motility, just naming a few. O-GlcNAcylation is heavily involved in our biology, and thus maintaining O-GlcNAc homeostasis inside our cells is of vital importance. When O-GlcNAc homeostasis is broken, when we have aberrant O-GlcNAcylation, this has been linked to a variety of different diseases, including but not limited to cancer, diabetes, cardiovascular, Alzheimer's, and Parkinson's disease. So O-GlcNAcylation and these two enzymes that cycle this modification are heavily involved in our biology. They've been linked to disease, so we're really interested in learning more about them. However, it's not that easy. There's a lot of challenges in studying these modifications. A primary one is that the substrate recognition of OGT is poorly understood. Now, there's a few reasons for this. One of them is that there's no conserved sequence motif for proteins that are O-GlcNAc modified. So you can't just look simply and identify a serine or threonine site on these proteins and know for certain whether or not it's O-GlcNAc modified. Additionally, full-length OGT is not amenable to structural characterization. We do know that OGT is a multi-domain protein, though, that contains 13.5 tetratricopeptide repeats and a catalytic domain that's interrupted by a unique fold intervening domain. Now, while full-length OGT isn't amenable to structural characterization, we do have a structural model by solving the crystal structure of OGT in two parts. First, in 2004, there was obtained a crystal structure of 11.5 of the TPRs, and then later in 2011, they solved the crystal structure of OGT 4.5, which contains 4.5 of the TPRs, the catalytic domains, and the intervening domain. So OGT 4.5 has 4.5 of the TPRs, the 11.5 TPR construct has 11.5, there's 13.5 total, so we get that nice overlap and we're obtaining this model that way. Additionally, I want to add that OGT 4.5 has been used quite extensively in studies, especially with peptide substrates. However, OGT 4.5, while it can glycosylate peptide substrates really well, it can't do that with full-length proteins, meaning that much of this TPR domain that's excluded in this OGT 4.5 construct, we know has some role in recognizing protein substrates. Additionally, many of the N-terminal TPR residues outside of this OGT 4.5, what their role could be in protein substrate recognition, or just in general their function, is not very well studied. So we don't really know what those residues do. And our understanding is actually so poor, there are multiple binding hypotheses on how OGT could be binding its substrates. One of the chief ideas that is floating around right now is that the protein substrate traverses the lumen, so the inside of this TPR domain, to extend into the active site. However, some people also think that potentially these proteins could interact with residues on the outside of this TPR domain. So currently, we know that the TPR domain is involved in protein substrate recognition, but we don't really know how it does that, and there's a lot we don't know about OGT itself. Now, you might be thinking, well, surely there should be a lot of techniques out there that we could use to study this. I briefly alluded to talking about substrate recognition, deconvoluting that through structural biology. You have your traditional binding assays, and then you have radiolabeled kinetics. However, each of these techniques struggle while studying OGT in some way, shape, or form. In the case of the structural biology approaches, there's no full-length OGT crystal structure. So that stops us from being able to generate a complex of OGT with a protein substrate. Then you can easily identify those residues and then do orthogonal studies to determine their exact role. So we can't do that, unfortunately, at the current time. And for cryo-EM, the N-terminus of OGT is trying to prove quite flexible. So even taking it at a cryo-EM approach is still very challenging. Considering the more traditional binding assays, such as SPR and ITC, OGT follows a bi-bi mechanism that's ordered. So there's two binding events. So first, OGT binds UDP-GlcNAc, so the sugar donor, and then it binds either the protein or peptide substrate. So these two binding events, SPR and ITC, they have a really hard time distinguishing between the two. And for considering radiolabeled kinetic, while that can get us a lot of useful parameters and we can learn a lot of information about it, it's very time consuming and it's not very efficient for rapid screening. So ideally, we can get something that's more high throughput, faster than radiolabeled kinetic, but can also still get us the information that we want and be able to effectively discriminate between sugar or protein, which is what we're very interested in. So to be able to address this issue in 2017, our lab developed GlycoNet electrophile probes, so GEPs. Particularly, we developed GEP1A. So this is a UDP-GlcNAc analog with two modifications that I want to point out. On carbon 6, we have this AZAC handle, which the functionality of that I'll get into in a minute. And what I want to focus here on is this N-acetyl has an allyl chloride functional group. Now this electrophile can covalently label a unique cysteine in the active site of OGT, and in the absence of a protein substrate, it can covalently label it. However, since it's a UDP-GlcNAc analog, when in the presence of a suitable substrate protein, GEP1A can be transferred onto the substrate protein to glycosylate it, and then the covalent labeling on OGT is quite minimal. And how this AZAC handle comes into play is that we can apply it for subsequent click chemistry through an alkyne fluorophore and then perform in-gel fluorescent detection to rapidly be able to see the relative labeling on OGT and the protein substrate. And ultimately, we can apply this to screen an array of OGT mutants quite rapidly. In treating it with a protein substrate in the GEP1A, undergoing click chemistry, subsequent in-gel fluorescent detection, we can get a nice predicted readout that allows us to rapidly and efficiently discriminate between protein binding and sugar binding. And to give you an example of what this would look like in these readouts, in the case of a mutant having impaired protein binding effect, the GEP1A still, when protein binding is impaired, is still able to covalently label OGT. So we expect OGT labeling to increase. However, since it can't bind a protein substrate, it's not going to glycosylate with the GEP1A, and that protein labeling, the substrate protein labeling, is going to decrease. However, contrasting from impaired sugar binding, similarly, the glycosylated protein, since the sugar, the GEP1A, is not in the active site, it's not going to be used to transfer and then glycosylate the protein. So that labeling is going to decrease. And also, since sugar binding is impaired, we're going to have decreased levels of OGT being labeled. So having this assay in hand, being able to rapidly screen these mutants, and then be able to discriminate between the protein and the sugar binding, that's all very exciting and that's great. But now we have to think about what kind of substrate protein we want to test. As I mentioned before, there's over 4,000 human proteins that are O-GlcNAc modified. So potentially that's a lot to choose from. So we want to narrow it down and think about what kind of substrate protein we want to test. So for studying the substrate recognition of OGT, we want to be choosing something that is mono O-GlcNAcylate. Many of OGT substrates have multiple O-GlcNAcylation sites throughout the protein. And if we're trying to map how OGT interacts with a substrate protein, we want it to be mono O-GlcNAcylated because having many O-GlcNAcylation sites would really confound our results, because one residue of OGT might be important for finding one residue or one part of this glycosylating one of the sites on the substrate protein, but not another. So having a single O-GlcNAcylation site is very important. We want a substrate that's more interesting, so it should be biologically relevant in some way. And of course, it needs to be something biologically recombinantly expressed. So it has to have high yield, high purity. Ultimately, you end up choosing the sole enzyme that reverses O-GlcNAcylation. So that's O-GlcNAcase or OGA. So that protein has a single O-GlcNAcylation site on serine 405, can be recombinantly expressed well, very biologically important. And also recent reports have shown that OGA purely coordinates with OGT. So we're really excited to choose OGA in our lab. It has all the criteria we're looking for, very easy to express, purify, and very interesting. So at this point, we select OGA as a protein substrate we want to test. And now we want to validate it against the GEP1A assay, making sure we can get the exact kind of readout that we're looking for here. And to do this, we tested against a negative control, so lacking OGT. We tested against OGT wild type, and then we tested against 3 different OGT mutants that we know what kind of function they're going to have. So in the case of the K842A mutant, that's going to have impaired sugar binding. This lysine is positively charged, and it interacts with the negatively charged phosphates that help stabilize the UDP-GlcNAc or the GEP1A staying inside the active site of OGT. So the K842A is going to have impaired sugar binding, so we expect decreased labeling on OGT and OGA. In the case of OGT 4.5, that's the truncated OGT that I mentioned before, and that one is missing 9 out of the 13.5 TPRs. So we expect to see decreased or impaired protein binding. So increased labeling on OGT, decreased labeling on OGA. And finally, in the case of the C917S, this is performed in the absence of substrate protein OGA. And this is just going to give us a general sense of the background level labeling on OGT in this assay. So we were really excited with these results. We see with the K842A, we see decreased OGT labeling, decreased OGA labeling, which is indicative of impaired sugar binding, which is exactly what we're looking for. In the OGT 4.5, we see increased OGT labeling, decreased OGA labeling, again, indicative of impaired protein binding. So again, exactly what we're expecting to see. In the C917S, we see a bit of background, but not too much, indicating that this assay is, at this point now, good to go, and we're good to test this against a series of OGT mutants that we want to investigate. Now, at this point, we have to decide what mutants we want to choose for studying OGT substrate recognition. We want to focus in on the TPR domain, since we know that that has a role in recognizing the substrates. However, the TPR domain has around 500 amino acid residues, so that's potentially a lot to choose from. So we have to think about which residues we want to choose. So we start off by choosing residues that are on the outside surface of the TPR domain, and some that are on the inside within the first few TPRs. And I want to add, when we're choosing these and other residues that I'll show in a second, we keep carefully in mind their positioning on the TPR domain. First, we want to make sure that they look like they could potentially interact with the substrate protein. And additionally, we're looking carefully at this crystal structure and making sure the residues don't have a series of hydrogen bonding or different kind of interactions with neighboring TPR residues, because those residues might be very vital for maintaining the 3-dimensional shape of the TPR domain. And we don't want to change the shape of the TPR domain because that would confound our results. So in addition to choosing the outside and these few N-terminal residues, we also chose to studied the full asparagine and aspartate ladder. While a few of these residues closer to the active site have been selected and studied, just a few of them, and they were found to be important, almost the entirety of the domain has not been studied. So we're really excited to select this full TPR domain to go ahead and screen. So before I show you all my results, I want to show you some representative data, which had some pretty exciting results that I want to share with you guys. Here, we're looking at a histidine and serine mutant. So these are the ones in the more N-terminal region. And we're also looking at a few of the asparagine mutants, so members of these asparagine ladder. And what we find in here is these few N-terminal residues and a couple of these asparagine ladder residues. There was no real significant change in the OGA or OGT label, meaning that those residues are likely, they're dispensable for the glycosylation of OGA. However, with this N220A, we see drastic decreases in OGA labeling and drastic increases in OGT labeling, meaning that this residue has some sort of role in OGT recognizing OGA, and we're seeing with this mutant impaired protein binding. So this is very exciting. And of the 30 mutants that we test, we find 14 with significant changes from the wild type OGT for both OGA and OGT labeling. And looking at this OGA labeling, many of these asparagines and some of these, and this aspartate here, show drastic decreases in OGA labeling. Additionally, we were excited to see that a few of these aspartates, a little closer to the active site here now, had increased labeling on OGA. Now looking at OGT labeling here for these same mutants, we see that those that had increases in OGA labeling, had decreases in OGA labeling, had drastic increases in OGT labeling, which is indicative of these mutants having impaired protein binding. However, in the case of these aspartates here, which had increases in OGA labeling, now have decreases in OGT labeling, which is indicative of these mutants having enhanced protein binding. So, in other words, these aspartates here that were kind of closer to the active site, we actually see that they can glycosylate OGA better than wild type OGT, which is really exciting to see. And to kind of look at this in a more 2D model, so you can get a better sense of how close these are and what their relative positionings are in the actual structure, we see that all the outside facing residues, and even these N-terminal ones, were dispensable for OGA glycosylation. Additionally, we found similar things for a few, but not all of the asparagine and aspartates. Many of these asparagine and aspartate ladder residues were functionally essential and played some sort of role in OGA glycosylation, including ones that were up to 10 nanometers away from the active site. And to look at this in a three-dimensional model so you can get a better sense of the kind of the location of these, we can see that the aspartates that I mentioned that were closer to the active site that actually these aspartates hinder OGA glycosylation are colored in this violet color. And all of these residues here line the inside of the TPR domain. And if we look down the lumen of the TPR domain, you can imagine this nice binding tunnel where OGA would occupy and lead directly into the active site of OGT to be glycosylated. So with these exciting results, we wanted to next validate these using radiolabeled kinetics. However, we don't want to choose all of the mutants because we tested 30, so that's just way too many to test with radiolabeled kinetics. So we chose residues, a few that were either further or closer to the active site, which is highlighted in green here, so you can tell where the active site is. And what we see is, as I selected these residues, that all of these mutants had impaired protein binding. As expected, we see a drastic decrease for them in the catalytic efficiency. Interestingly, for the ones that were further away, so this D152A and the 155, we see drastic increases in the KM, but not real changes in the turnover number. So this is further supporting that these residues are primarily involved in the protein binding aspect and facilitating the interaction between OGT and OGA in that manner. However, with the residues that were a little closer to the active site, they have less drastic increases in the KM, and we do see notable decreases in the KCAT, which gives us the impression that, yes, these residues indeed are still functionally important for OGT recognizing OGA through the protein binding mechanism. However, there might be something more going on with these residues here, such as potentially maybe they alter the positioning of the OGA in the active site, and then that in itself is changing some of the rates, maybe the rate of sugar transfer, or the release of OGA binding OGT, or part of the release. So at this point, we understand how OGT is recognizing OGA, and that's very interesting, but we don't really understand how OGA itself, what regions of OGA might be important for it being recognized by OGT. We do know that OGA, like OGT, is a multi-domain protein. However, OGA, it contains a catalytic domain, a stalk domain with a disordered loop where the glycosylation site happens, and it also contains a pseudohistone acetyltransferase domain. Now, this N-terminal disordered region and the pseudohistone acetyltransferase domain, they have a known function. So we don't really know what those regions do. So we're really interested in knowing whether those regions could play some sort of role in OGA being glycosylated. Maybe these regions are interacting with OGT, and that allows OGA to be glycosylated. And since I see we're running a little late on time, I'll just kind of summarize this data here by saying that what we found is these two regions were dispensable for the glycosylation of OGA, so that OGT primarily bound to the catalytic domain and the stalk domain of OGA, and by deleting the HAT domain and N-terminal region, we didn't see any notable difference in the glycosylation patterns. So skipping through this to save some time. So in summary, what we found, we identified of the 30 mutants that we tested, 14 of them that functionally impact OGA glycosylation, spanning almost the entire length of the TBR domain, going out 10 nanometers away. And this study here represents the first systematic test where we actually look at each of these TBR domains and look at residues from each of them. These results support the inside binding hypothesis for OGA, meaning that the residues span the inside of the TBR domain, not the outside. And also we found that the HAT domain and N-terminal region of OGA are dispensable for its glycosylation. And I'd also like to add, very recently after we published these results, the Walker lab actually published results looking at a few different protein substrates than what we looked at. And they also saw that some of the further out residues were also important for glycosylating a few other proteins, thus supporting our finding and adding on to this general picture of what we have for OGT substrate recognition, which is coming along really nicely. We're getting a much more clear idea of how OGT is recognizing its substrates. And what's really exciting to learn more about OGT substrate recognition, and we hope to potentially be able to develop new assays, new studies on how we can potentially further study OGT and O-GlcNAcylation, knowing more about how OGT recognizes its substrates. And with that, of course, I'd like to thank all my committee members, especially my boss, Dr. Jiaoyang Jiang, all of my lab members, especially Da-Chang and Ao, who did the radiolabeled Kinetics for these experiments, Hao and Matt, who synthesized GAP1A, funding sources, the university and NIH, and also all of you guys here for coming today, and the organizers for this seminar series, which I've been attending recently. It's been a real treat to attend, and a real honor to be able to present. And with that, I'd like to take any questions.